Take a moment to appreciate how spectacularly unhelpful that definition is. A self-sustaining chemical system capable of Darwinian evolution. That's what 40 years of astrobiology, billions of dollars in research funding, and the combined intellectual output of thousands of the smartest people in human history has produced as an answer to the question, what is life? A sentence that also technically describes a lit candle, or a wildfire, or a crystal growing in a supersaturated solution, or, depending on your philosopher, the stock market. The definition is not wrong, it's just breathtakingly circular. We defined life by listing what the only life we've ever found does. We looked at carbon-based, water-dependent, DNA-using organisms on one rocky planet orbiting one unremarkable star and said, that's the template, that's the benchmark, go find more of that. And then we sent our probes to other worlds specifically looking for the chemistry we already understood, looking for water, looking for carbon, looking for amino acids, looking for the molecular signatures of life that is, essentially, a variation of us. Here's the problem. In 1976, the Viking landers touched down on Mars and ran a suite of biology experiments looking for chemical signs of life. One experiment, the labeled release experiment, returned a positive result. Something on Mars was metabolizing. Something was responding to the test in exactly the way a living organism would respond. NASA declared it a false positive, an unusual chemistry, not life. Case closed. The scientist who designed the experiment, Dr. Gilbert Levin, spent the rest of his life arguing they were wrong. What if he was right? What if we found life on Mars in 1976, declared it dead on arrival because it didn't look carbon-friendly enough, and walked away from the most important discovery in human history because our definition of life was too small? This is non-carbon life, the aliens will never recognize. And by the end of this, you'll understand why the most dangerous assumption humanity has ever made isn't about weapons or politics or artificial intelligence. It's about chemistry. Let's start with why carbon is so absurdly, almost suspiciously good at being the foundation of life. Because to understand why anything else might work, you first need to appreciate just how much carbon has going for it. Carbon is, without question, the most overachieving element in the periodic table. It's the kind of element that would be insufferable at parties if elements had personalities. Carbon has 4 valence electrons. This means it can form 4 chemical bonds simultaneously. 4, not 2 like oxygen, not 1 like hydrogen. 4. This might sound like a minor technical detail. It is not. It is the entire ballgame. With 4 bonding slots, carbon can chain to other carbon atoms indefinitely, forming long, stable backbones. It can form rings. It can form branching structures. It can form double bonds and triple bonds that introduce rigidity and reactivity at specific points. It can bond to hydrogen, oxygen, nitrogen, sulfur, and phosphorus simultaneously, creating the staggering chemical diversity of organic molecules. There are over 10 million known carbon-based compounds. For comparison, silicon, which we'll get to, has around 100,000 known compounds. Carbon doesn't just participate in chemistry. Carbon is chemistry's overachieving parent who volunteered for every school committee and somehow built the entire gymnasium by themselves on a Tuesday. The reason life as we know it is carbon-based isn't some cosmic accident or arbitrary preference. Carbon makes genuinely extraordinary molecules. DNA, the information storage molecule of all known life, is a carbon polymer. Proteins, the workhorses of cellular function, are carbon chains, folded into impossibly precise 3-dimensional shapes. Cell membranes are made of carbon-based lipids that spontaneously form bilayers in water, creating enclosed compartments where chemistry can happen in protected isolation. That last part is worth emphasizing. Certain carbon molecules, when placed in water, will spontaneously organize themselves into cell membrane-like structures. No intelligence required, no instruction,
just carbon chemistry doing what carbon chemistry does when mixed with water. It self-organizes. It's almost as if carbon and water were designed for each other. Water is the other half of this partnership. Water is the universal solvent. It dissolves more substances than virtually any other liquid. It remains liquid across a relatively wide temperature range, 0 to 100 degrees Celsius at standard pressure. It has high surface tension and excellent heat capacity, stabilizing temperature against rapid swings, and its hydrogen bonding properties create a molecular scaffold that proteins and DNA exploit for their three-dimensional structures. Carbon and water, together, appear to be the ultimate team for building complex, self-replicating, evolving chemistry. We found this combination producing life from the deepest ocean trenches to the upper atmosphere, from volcanic vents to Antarctic ice sheets, from pH 0 acid pools to pH 13 alkaline lakes. Carbon and water life is so adaptable, so resilient, so chemically creative that it has colonized essentially every environment on Earth. And here's where the assumption bakes itself in. Because this chemistry works so well, and because it's the only chemistry we've ever personally observed producing life, the entire framework of astrobiology, the scientific search for life beyond Earth, has been constructed around finding it again. The working hypothesis, explicitly stated in most mission parameters, is follow the water, look for carbon, search for biosignatures that carbon-based life would produce. These have driven billions of dollars in mission design. The Curiosity and Perseverance rovers on Mars both carry organic chemistry detectors. The Europa Clipper is specifically tuned to detect water-soluble organic compounds. The James Webb Space Telescope's atmospheric analysis targets oxygen, methane, and CO2, gases that carbon-based life would produce. These are not wrong priorities. If carbon and water life exists on Mars or Europa, these instruments will find it. The problem is what they'll miss, specifically, everything else. Because the universe does not, as far as anyone has established, have a contractual obligation to use carbon and water as its medium of choice for self-organizing chemistry. The universe is 13.8 billion years old. It contains an estimated 2 trillion galaxies, each with hundreds of billions of stars, each frequently orbited by planets. The conditions on those worlds range from liquid methane lakes to sulfuric acid clouds to surfaces hot enough to melt lead to environments with no solvent liquid at all, just gases and plasma. Carbon and water life would not survive most of these environments, but the question isn't whether carbon and water life can survive there. The question is whether the universe found a different solution entirely. Whether in 4 billion years of chemistry happening on a hot, sulfurous, methane-soaked world, something began to self-replicate using completely different building blocks. The most obvious alternative is sitting directly beneath carbon on the periodic table, 4 rows down, 4 extra protons in the nucleus, wearing the same number of valence electrons, like a slightly less impressive costume. Silicon. Chemically speaking, it's carbon's understudy, and like every understudy in history, it's been waiting for its moment.
Silicon is already the second most abundant element in Earth's crust, behind oxygen. You are literally standing on it. The ground beneath you is mostly silicon dioxide, quartz, sand, rock, and silicon has been building complex mineral structures for 4.5 billion years, quietly, without asking for any credit. Here's what makes silicon appealing as an alternative basis for life. Like carbon, it has 4 valence electrons. Like carbon, it can form 4 simultaneous chemical bonds. Like carbon, it can chain to other silicon atoms, forming silicon-silicon bonds that could theoretically serve as a molecular backbone. Like carbon, it can bond to oxygen, hydrogen, nitrogen, and a variety of other elements. The structural similarities are real and not trivial. And silicon chemistry is not entirely boring. Silicones, the polymers you know from medical implants, cookware coatings, and the contents of various cosmetic products, are silicon-oxygen chain molecules with remarkable stability, flexibility, and resistance to heat and oxidation. Silicates form enormous diversity of crystal structures. Some silicon compounds form ring and cage structures with genuine complexity. In 2016, scientists at Caltech achieved something remarkable. They engineered bacteria, specifically E. coli, to produce carbon-silicon bonds inside living cells. Biology creating organosilicon compounds, which had previously only been made through industrial chemistry. Life, slightly nudged, willingly started incorporating silicon into its chemistry. The lead researcher, Francis Arnold, won the Nobel Prize in Chemistry in 2018 for related work. The result demonstrated that the barrier between carbon-based biology and silicon-incorporating chemistry is not a wall, it's a gradient, not an either-or, a spectrum. So why isn't silicon the dominant element of life on Earth? Why did carbon win? Because silicon has some serious fundamental problems, and they're worth understanding because they define the environments where silicon-based life might work, even if it can't work here. Problem 1. What silicon does when it oxidizes? When carbon bonds with two oxygen atoms, you get carbon dioxide, CO2, a gas, water-soluble, breathable. At room temperature, you can exhale it, and plants can absorb it, and the whole carbon cycle functions because the oxidized waste product of carbon metabolism is a gas that moves freely through the environment. When silicon bonds with two oxygen atoms, you get silicon dioxide, SiO2, quartz, rock, solid at room temperature. If silicon-based life metabolized silicon the way we metabolize carbon, it would constantly be filling itself up with sand. Its waste product would be glass. This is less than ideal from a biological standpoint. Imagine if every time you exhaled, instead of releasing carbon dioxide, you deposited a small rock inside your lungs. That's what silicon-based biochemistry runs into at Earth temperatures. The oxidized byproduct doesn't leave, it stays, it accumulates, it turns you into a geological event. However, here's where it gets interesting. At extremely high temperatures, hundreds to thousands of degrees Celsius, silicon dioxide becomes volatile. At those temperatures, the chemistry changes entirely. The problems that make silicon-based life impossible at room temperature start to dissolve, and silicon's advantages start to matter. On a world with surface temperatures above 500 degrees Celsius, a volcanic hellscape by Earth standards, perfectly mundane by the standards of many exoplanets, silicon chemistry might behave differently. Silane compounds, silicon bonded to hydrogen, analogous to hydrocarbons, are more stable at those temperatures. Silicon-oxygen chemistry produces gases rather than solids. The whole landscape of what's possible shifts. Venus has a surface temperature of around 465 degrees Celsius. Its atmosphere is thick with sulfuric acid. Nothing carbon and water-based could survive on the Venusian surface for more than seconds. But for silicon-based chemistry, the temperature is moving into potentially workable territory. There are exoplanets called super-Earths, with surfaces maintained at high temperatures by internal heat, tidal forces, or proximity to their stars. Lava worlds, volcanic archipelago worlds, worlds where surface temperatures are high enough that silicon chemistry operates in the regimes where it starts looking more competitive against carbon. But silicon has a second problem, and it's trickier. Carbon-based life uses water as its solvent, the medium in which all its chemistry happens. Silicon is not water-soluble. You can't dissolve silicon compounds in water and expect the kind of dynamic, fluid biochemistry that makes carbon-based life possible. Silicon needs a different solvent. Candidates include liquid sulfur, stable at around 120 to 450 degrees Celsius,
or sulfuric acid, which remains liquid across a similar range, or ionic liquids, salts that are liquid at room temperature and can dissolve a remarkable range of compounds, or supercritical carbon dioxide, which exists above 31 degrees Celsius at sufficient pressure and behaves like a cross between a liquid and a gas with extraordinary solvent properties. Io, Jupiter's innermost large moon, has surface temperatures ranging from -143 Celsius away from vents to 1600 Celsius at volcanic eruption sites, with sulfur dioxide frost, liquid sulfur flows, and sulfur dioxide gas atmospheres. It is, by every carbon-centric standard, biologically dead. But as a chemistry environment for silicon sulfur-based life, it's a different conversation. We have never looked for silicon-based biosignatures on Io. We haven't designed instruments tuned to detect them. We've looked at Io and seen a hellscape and moved on. We're doing the astronomical equivalent of walking into a store, not finding the specific item we wanted, and leaving without asking if they have it in the back. And now, silicon is just the beginning, because the exotic biochemistry zoo gets considerably stranger from here. Silicon compounds generally react more slowly than carbon compounds at equivalent temperatures. Silicon chemistry is sluggish at Earth temperatures, another reason carbon dominated here. But slow chemistry is not dead chemistry. It's chemistry operating on a different clock. Carbon-based life on Earth operates on timescales of microseconds to decades. A silicon-based organism on a cool world might think in millennia, react in centuries, live for geological eons. From our perspective, it would look like particularly interesting rock formation. We would core sample it for geological data, contaminate it with drill lubricant, and fly home satisfied that we'd found no life. And now, silicon is just the beginning, because the exotic biochemistry zoo gets considerably stranger from here.
Silicon is
Titan
already the
is
second most
Saturn's
abundant
largest
element
moon.
in Earth's
It
crust,
has a
behind
thick
oxygen.
nitrogen
You are
atmosphere,
literally standing
thicker
on it.
than
The
Earth's.
ground beneath you is
It has
mostly
weather
silicon
systems,
dioxide,
rain,
quartz,
rivers,
sand,
lakes,
rocks, and silicon has
seas.
been building complex
It has a
mineral
complete
structures for
hydrological
4.5 billion
cycle.
years.
The liquid collects
Quietly,
on the surface,
without asking for
evaporates,
any credit.
forms clouds,
Silicon is
and
already the
rains back
second
down.
most abundant element in
Here's the
Earth's
catch.
crust, behind
The
oxygen.
liquid isn't
You are
water.
literally standing
It's
on
methane.
it. The ground beneath
Liquid
you is
methane
mostly silicon
at
dioxide,
-179
quartz,
degrees Celsius,
sand,
pooling
rock,
in vast hydrocarbon
and
seas at the
silicon
poles,
has been building
raining
complex
down onto
mineral
a landscape
structures
of
for
water, ice
4.5
rocks,
billion
and organic
years.
sand dunes.
Quietly,
Titan's
without
rivers
asking
are
for any
methane
credit.
rivers.
Here's what makes
Its
silicon
lakes are
appealing as an
methane
alternative
lakes.
basis for
Its
life.
rain
Like
is
carbon,
methane rain.
it has 4 valence electrons.
NASA is
Like
sending
carbon,
a
it can
nuclear-powered
form 4
rotorcraft
simultaneous
called
chemical
Dragonfly
bonds.
to Titan
Like carbon,
in the late
it can chain
2020s.
to other silicon
They are very
atoms,
excited
forming
about Titan.
silicon-silicon bonds
Titan
that
is
could
considered one
theoretically
of the
serve as a
best
molecular
places in the solar
backbone.
system
Like
to look for
carbon,
life.
it can bond to
And yet
oxygen,
almost every
hydrogen,
press release
nitrogen,
about
and a
Titan
variety
life
of other elements.
qualifies it
The
with
structural
not
similarities
life
are
as we know
real
it.
and not trivial.
Because
And
we're looking
silicon
for life
chemistry
that
is
needs
not
water.
entirely boring.
Meanwhile,
Silicones,
Titan has
the
methane
polymers
lakes,
you know from
and we're
medical
like,
implants,
probably
cookware
dead,
coatings,
lol.
and the contents of various
But
cosmetic
here's the
products
scientific
are
case
silicon
for
oxygen
ammonia-based
chain
life,
molecules with
which is the
remarkable
leading
stability,
candidate for Titan-type
flexibility,
environments.
and resistance to heat
Ammonia,
and oxidation.
NH3,
Silicates
shares
form
several
enormous
important
diversity of
properties with
crystal
water.
structures.
It's a
Some
polar
silicon
solvent,
compounds
meaning it
form
can
ring and
dissolve
cage
a
structures
wide
with
range of
genuine
ionic and
complexity.
organic
In
compounds.
2016,
It has a
scientists at
relatively high
Caltech
heat
achieved something
capacity.
remarkable.
It can
They engineered
participate in
bacteria,
acid-base chemistry.
specifically E.
It
coli,
remains liquid
to produce
across a
carbon
temperature
silicon
range
bonds
of
inside living
-78
cells,
to
biology creating
-33 degrees
organosilicon
Celsius at
compounds,
standard
which had
pressure,
previously only
which
been made
means
through
on
industrial
Titan,
chemistry.
where it could be mixed with
Life,
methane and other
slightly
hydrocarbons,
nudged,
it
willingly
might serve
started
as a component
incorporating
of a
silicon
complex
into its
liquid
chemistry.
solvent
The
system.
lead researcher, Francis
Ammonia
Arnold,
water
won the
mixtures
Nobel
have
Prize
even
in
more
Chemistry in
interesting properties.
2018 for
At certain
related work.
ratios,
The result
mixture has a
demonstrated that the
significantly
barrier between
depressed freezing
carbon-based
point,
biology
staying
and
liquid
silicon
at
incorporating
temperatures far
chemistry
below
is not a
0
wall.
Celsius.
It's a gradient,
In a
not an
subsurface
either-or,
ocean on
a
Titan
spectrum.
or similar
The lead
moon,
researcher,
where ammonia,
Francis Arnold,
water,
won the
and
Nobel
methane
Prize in
coexist
Chemistry
under
in
pressure.
2018
You
for
might have a
related
liquid
work.
medium
The
with
result
genuinely
demonstrated that the
interesting
barrier
solvent
between
properties for
carbon-based
complex
biology
chemistry.
and silicon incorporating
Researchers,
chemistry is
including
not a wall.
astrobiologist David Schultz
It's against
McCooch
a
and chemist
gradient,
Robert
non
Shapiro,
have proposed that Titan's
Not
hydrocarbon
ada
lakes
they
could
can now
host
or yada
life
either
using
or.
acetylene and hydrogen as an energy
A
source.
spectrum.
A metabolic
So why isn't
pathway
silicon the
completely
dominant
alien to
element of
any
life on
Earth
Earth?
organism.
Why did carbon
Instead
win?
of photosynthesis,
Because silicon has some
instead of
serious
hydrothermal
fundamental
vent
problems,
chemistry,
and they're worth
imagine an
understanding
organism
because
that
they define the
breathes
environments
hydrogen
where
and
silicon-based
eats
life
acetylene
might work,
dissolved in
even if
methane.
it can't work here.
The waste product
So
would
why
be
isn't
methane.
silicon the
And here's the
dominant
remarkable
element of
thing.
life on Earth?
Cassini's data
Why did
on
carbon
Titan's
win?
atmosphere
Because
showed an
silicon
anomalous
has some
depletion of
serious
hydrogen near the
fundamental
surface
problems, and
and
they're
the
worth
depletion of
understanding.
acetylene on
Because
the
they
surface
define the
compared to
environments where
atmospheric expectations.
silicon-based life might
Both
work,
of these
even
depletions
if
are
it can't
exactly
work
what
here.
you'd expect
Problem
if
1.
something were
What
consuming
silicon does
hydrogen
when it
and acetylene
oxidizes.
at the
When
surface.
carbon bonds with two
Not proof,
oxygen atoms, you
but a
get carbon
pattern that doesn't
dioxide,
have a satisfying
CO2, a gas,
non-biological
water-soluble,
explanation.
breathable.
NASA's
At room
response was
temperature, you
measured
can
and
exhale
cautious
it, and plants
and
can
eminently
absorb
sensible,
it. And the whole
and
carbon
also
cycle functions
spectacularly
because the oxidized
unsatisfying.
waste product
There
of
could
carbon
be a
metabolism
non-biological
is a gas
explanation.
that moves freely
Yes,
through the
there could
environment.
be.
When
There could
silicon
also be
bonds with
tiny
two oxygen atoms, you
methane-drinking
get
organisms,
silicon
and
dioxide,
we're flying a rotorcraft
SiO2,
to find out,
quartz, rock,
which, at least is
solid
something.
at room temperature. If
Now, let's
silicon-based
talk about
life
sulfur.
metabolized
Specifically,
silicon,
let's
the way
talk
we
about life
metabolize
based on
carbon,
sulfur
it would
chemistry
constantly
in
be filling
extreme
itself up with
environments.
sand. Its
Sulfur
waste
is
product would be
already
glass.
deeply embedded
This is
in
less
Earth
than ideal
biology.
from a
It's
biological
present in
standpoint.
2 amino
Imagine if
acids,
every time you
cysteine
exhaled,
and
instead of
methionine,
releasing
and
carbon
plays critical
dioxide,
structural
you
roles in
deposited a
many
small
proteins.
rock inside your
But
lungs.
sulfur's
That's
potential
what
as an
silicon-based
alternative
biochemistry
biochemical
runs
foundation
into at
goes
Earth temperatures.
considerably further
The
than
oxidized
being a minor
byproduct doesn't
participant
leave.
in
It
carbon-based
stays.
life.
It accumulates.
Venus's
It turns
cloud
you into a
layer, at
geological
altitudes
event.
of 48
However,
to 60
here's where it gets
kilometers,
interesting.
have
At
temperatures
extremely
between
high
30 and
temperatures,
70 degrees
hundreds to
Celsius,
thousands of degrees
comfortable
Celsius,
for Earth
silicon
organisms.
dioxide
It has
becomes
abundant
volatile.
sulfur
At
dioxide
those
and
temperatures, the
sulfuric
chemistry
acid.
changes entirely.
pressure is
The
manageable,
problems that make
sunlight is
silicon-based
available.
life impossible
It
at
is,
room
in
temperature start to
atmospheric
dissolve,
terms,
and silicon's
genuinely
advantages
hospitable
start to matter.
for
On a
something
world
that
with
can
surface
tolerate
temperatures above
sulfuric
500
acid as
degrees
its medium.
Celsius, a volcanic
On Earth,
hellscape by Earth
organisms
standards,
called
perfectly mundane by the
acidophiles
standards of
thrive
many
in
exoplanets.
pH-0 environments.
Silicon
Sulfuric acid
chemistry
concentrations that
might
would dissolve
behave
most materials.
differently.
Ferroplasma
compounds,
acidomarnis,
silicon
a
bonded
microbe
to
discovered in
hydrogen,
the iron
analogous
mine
to
county in
hydrocarbons,
California,
are
lives
more
happily
stable
in
at those
pure
temperatures.
sulfuric acid solution.
Silicon oxygen
These
chemistry
organisms
produces
didn't evolve
gases
for this
rather than
environment by
solids.
slowly adapting.
The whole
They
landscape
are
of
simply
what's
operating
possible
at the
shifts.
extreme end
On a
of what
world
carbon
with surface
and water
temperatures
chemistry
above
allows.
500 degrees
A
Celsius,
sulfur-based
a volcanic
biochemistry
hellscape
native
by
to a
Earth standards,
sulfuric acid-rich
perfectly mundane
environment
by the
wouldn't
standards
need
of
to
many
tolerate
exoplanets.
acid. It would be built for acid. Sulfur
Silicon
bonds can
chemistry
form complex
might behave
polymeric
differently.
structures.
Silane
Sulfur can take
compounds,
on multiple
silicon
oxidation
bonded
states,
to hydrogen,
allowing it to drive
analogous to
redox chemistry.
hydrocarbons, are
The
more
transfer of
stable at those
electrons that
temperatures.
powers metabolism
Silicon
in all
oxygen
known
chemistry
life.
produces
A
gases rather
sulfur-based
than
metabolic
solids.
cycle
The whole
in a
landscape
world
of what's
saturated
possible
with
shifts.
sulfur compounds
Venus has a
might
surface
be
temperature of
functionally
around
analogous to the
465
carbon-based
degrees
metabolic
Celsius.
cycles of
Its
Earth
atmosphere
life,
is thick
just
with
written
sulfuric
in a
acid.
completely
Nothing
different
carbon and
alphabet.
water-based could survive
But
on the
silicon,
Venusian surface
ammonia,
for more than
and
seconds.
sulfur
But
are
for
all still
silicon-based
playing it
chemistry,
relatively
the temperature
conservative.
is moving into
They are at
potentially
least
workable
recognizable chemistry
territory.
using
Venus
elements
has
we
a surface
use,
temperature of
operating by
around
principles we
465
understand.
degrees Celsius.
Now we're going to talk
Its
about
atmosphere
two
is thick
proposals
with
that are
sulfuric
genuinely
acid.
structurally
Nothing carbon and
mind-bending.
water-based could survive on the
Proposals that
Venusian
challenge
surface
not
for
just
more
our
than
chemistry
seconds.
assumptions, but our
for
assumptions about
silicon-based
what
chemistry,
physical substrate
The
life
temperature
requires.
is moving into potentially
First,
workable territory.
plasma-based life.
There are exoplanets
Plasma
called
is the
super-Earths
fourth
with
state of
surfaces
matter,
maintained at high
ionized
temperatures
gas,
by internal
where
heat,
electrons have
tidal
been stripped
forces,
from
or
atoms
proximity
to
to their
create a
stars.
soup of
Lava
charged
worlds,
particles.
volcanic
Plasma is
archipelago
actually
worlds,
the most
worlds where
common
surface
state of
temperatures
matter
are
in the
high enough that
universe.
silicon
Stars
chemistry
are plasma,
operates in
the interstellar
regimes
medium
where it
is
starts
largely
looking more
plasma.
competitive against
The
carbon.
universe
There are
is
exoplanets
dominated by a
called
state of matter
super-Earths,
that
with
by our
surfaces
current
maintained at high
definition
temperatures
cannot
by
host
internal
life.
heat, tidal forces,
But in
or
2007,
proximity to their
researchers
stars.
Vadim
Lava
Sikowitz
worlds,
and colleagues
volcanic
published a paper
archipelago
in the New
worlds,
Journal of Physics
worlds where surface
demonstrating
temperatures
something
are high enough that
unexpected.
silicon
In
chemistry
laboratory
operates
plasma
in the regimes
conditions
where it starts
simulating the
looking more
dust plasma
competitive against
environment of
carbon.
space,
But
they
silicon
found
has a
that
second
electrically
problem, and it's
charged dust
trickier.
particles in
Carbon-based methane-based
plasma,
life uses water as its
spontaneously
solvent,
organized
the
into
medium in
helical
which all its
structures.
chemistry happens.
These structures
Silicon
could
is
store
not water
information in their
soluble. You
configuration,
can't dissolve
like a
silicon
physical
compounds
memory.
in water
They
and
could
expect the
replicate,
kind of
producing
dynamic
daughter
fluid
structures with
biochemistry
similar
that makes
configurations.
carbon-based
They
life
could
possible.
evolve,
Silicon
with different
needs a
variant
different
structures
solvent.
being more
Candidates
or less
include
stable
liquid
under
sulfur,
changing conditions.
stable at around 120
The
to
structures weren't alive.
450 degrees
Nobody
Celsius,
claimed they were,
or
but
sulfuric
they
acid,
exhibited
which remains
information
liquid
storage,
across a similar
replication,
range,
and selection.
or ionic
Three of the
liquids,
core
salts
properties
that
we
are liquid at
associate
room
with life,
temperature
using
and can
nothing
dissolve
but
a remarkable range of
electromagnetic interactions in
compounds, or
ionized gas,
supercritical carbon
without
dioxide,
without
which exists
water,
above 31
without
degrees
chemistry in the
Celsius at
traditional
sufficient
sense.
pressure, and behaves
Now
like a
take that
cross
principle and
between
put it in a
liquid
stellar
and a
atmosphere.
gas.
A
With
star's outer
extraordinary
layers
solvent
are
properties.
complex plasma
Io,
environments
Jupiter's
with
innermost
enormous
large moon,
energy throughput,
has surface temperatures
enormous
ranging
chemical,
from negative
or rather
143
electromagnetic
Celsius
diversity,
away from vents
and
to
billions of years of
1,600
time.
Celsius
The
at
question
volcanic
nobody
eruption
has
sites,
formally modeled
with sulfur
is,
dioxide
given
frost,
sufficient
liquid
time
sulfur
and energetic
flows, and sulfur
complexity,
dioxide
can
gas
self-replicating
atmospheres. It is, by
electromagnetic
every
structures
carbon-centric
emerge in
standard,
plasma?
biologically dead.
We don't know.
But as a chemistry
We haven't tried to
environment
find
for
out
silicon
because
sulfur-based
plasma-based life
it's a different
isn't in
conversation.
anyone's mission
Io,
parameters. It
Jupiter's
doesn't fit the
innermost large
definition.
moon,
It
has
doesn't
surface
make
temperatures ranging
biosignatures
from
we know
negative
how to look for.
143
So we
Celsius
look
away
at the
from
sun,
vents
a
to
4.5
1,600
billion-year-old
Celsius at
object
volcanic
of
eruption
extraordinary
sites, with sulfur
electromagnetic
dioxide
complexity,
frost,
and we
liquid
study it
sulfur
for physics,
flows, and
and
sulfur
we don't ask
dioxide
the
gas
biology
atmospheres.
question.
It is, by
Second,
every carbon-centric
mechanical
standard,
life, or more
biologically
precisely,
dead. But as a chemistry
self-replicating
environment for
non-biological
silicon
systems.
sulfur-based life,
In
it's a different
1948,
conversation.
mathematician
We have
John
never
von
looked for
Neumann
silicon-based
formally described a
biosignatures on
theoretical
Io.
construct
We haven't designed
now called
instruments
a von
tuned to
Neumann
detect
machine,
them. We've looked
a
at
device
Io and seen a
capable of
hellscape and moved
self-replication
on.
using
We're
available
doing the
raw
astronomical
materials.
equivalent
Von
of walking
Neumann
into a
mathematically
store,
proved
not
that
finding
such
the
machines
specific
were
item we
possible.
wanted,
He
and
did not
leaving
build
without
one,
asking if
but
they
he
have it in the
described
back.
in
And
rigorous
here's the
mathematical
final
terms
humbling thing
a
about
non-biological
silicon-based life.
system capable
Silicon
of the most
compounds
fundamental
generally
property
react
of life,
more slowly than carbon
reproduction.
compounds at
A
equivalent
von Neumann
temperatures.
machine doesn't
Silicon
need
chemistry
carbon.
is sluggish
It
at Earth
doesn't need
temperatures.
water.
Another
It
reason
needs raw
carbon dominated
materials
here.
and an energy
But slow
source
chemistry
and a
is not
sufficiently
dead
complex
chemistry.
starting
It's
design.
just chemistry
It
operating
could,
on
in
a different
principle,
clock.
operate on any
Carbon-based
world
life
with
on Earth
accessible
operates on
minerals and
timescales
usable
of
energy,
microseconds
which
to
describes
decades.
most
A
solid
silicon-based
planets and
organism
moons
on
in
a
the
cool
universe.
world might think in
This
millennia,
raises a question
react
that
in
most biology
centuries,
textbooks
live
decline to
for
engage with.
geological
If a
eons,
self-replicating
reproduce
machine
once every
satisfies the
million
reproduction
years.
criteria
From
for
our
life,
perspective,
and
it
if
would
can
look
be
like
modified by
particularly
external
interesting
forces,
rock
and
formation.
therefore
We would
undergo
core
selection,
sample it for
and if
geological
it
data,
maintains
contaminate it
internal
with drill
processes to
lubricant,
sustain
and fly
its
home,
operation, does
satisfied
it
that we
qualify
found
as
no
life?
life. And now,
Under NASA's
silicon
definition,
is just the
arguably
beginning,
yes.
because the exotic
Under most people's
biochemistry zoo
intuitive
gets
definition,
considerably
definitely
stranger
no.
from here.
And that discomfort is productive because it forces the question, what are we actually looking for? Are we looking for chemistry that resembles ours, or are we looking for the property, self-organized, self-sustaining, evolving complexity that we associate with life, regardless of what it's made of? These aren't idle philosophical games. They have immediate, practical consequences for every astrobiology mission currently flying or in development.
July 20th, 1976. The Viking 1 lander touches down on Chryse Planitia on Mars. It is the first successful soft landing on another planet. It carries a biology laboratory, 4 experiments specifically designed to detect signs of life in the Martian soil. It is, to date, the only time in human history that a spacecraft has directly tested the soil of another planet for biological activity. The labeled release experiment worked like this. A small amount of Martian soil was wetted with a nutrient solution containing radioactive carbon. If anything in the soil was metabolizing, consuming the nutrients, it would release radioactive carbon dioxide, which the instrument would detect. You'd see a spike on the readout indicating biological activity. The readout spiked, dramatically, immediately. The soil was producing radioactive CO2 at a rate consistent with biological activity. When the soil was heated to sterilizing temperatures, which kills microorganisms, the signal dropped. When fresh nutrient solution was added, the signal didn't spike again, which is unusual for a microbial response. NASA's conclusion: not life. The reasoning centered on the gas chromatograph mass spectrometer, a separate instrument that detected organic compounds, or rather, didn't. The GCMS found no organic carbon in the Martian soil. Since carbon-based life requires organic carbon, and since the soil apparently had none,
The labeled release result must be a chemical reaction, not a biological one. The favored explanation was that Martian soil contains perchlorates, highly oxidizing compounds that can produce the observed result chemically without life. And Mars does have perchlorates. We've confirmed that subsequently. Case closed, apparently. Except the GCMS wasn't designed to detect the specific organic compounds that Martian life, adapted to a perchlorate-rich environment, might produce. If Martian life incorporates perchlorates into its chemistry, as some Earth extremophiles do with otherwise toxic compounds, its organic molecules might be different enough from standard Earth organics that the GCMS would miss them. Dr. Levin spent over 40 years pointing this out. He noted that every positive control test, tests using known Earth microorganisms, produced the same pattern as the Martian result. He noted that the GCMS sensitivity threshold was inadequate for the Martian soil composition. He noted that the chemical explanation required a combination of conditions on Mars that had never been demonstrated to produce the exact observed signal. In 2008, the Phoenix lander confirmed perchlorates in Martian soil. The mainstream community treated this as vindication. See, chemical explanation confirmed. Levin treated it as exactly the opposite. Life adapted to a perchlorate-rich environment would use perchlorate chemistry. The labeled release result was entirely consistent with perchlorate metabolizing organisms. The debate is not resolved. It is merely ignored, because proceeding as if Mars is lifeless is more tractable than proceeding as if Mars might harbor chemistry-based life we can't fully interpret. Probably not life became definitely not life, became we're looking for different life now, without anyone formally closing the original question. And this is the recognition problem. Not just can we detect non-carbon life, but even with carbon-based life on a world right next door, running experiments in the 1970s, we may have gotten the answer and talked ourselves out of believing it. The biosignatures that modern missions search for are overwhelmingly the biosignatures of Earth-like carbon-based water-dependent life, oxygen in atmospheres produced by photosynthesis, methane alongside oxygen, unstable combination requiring replenishment, phosphine produced by anaerobic bacteria, complex organic molecules of specific structural types. What would silicon-based life produce as atmospheric biosignatures? Potentially different silane compounds, unusual silicon dioxide distributions, specific isotopic ratios of silicon across a planet's surface. None of our current atmospheric analysis instruments are tuned to interpret these patterns as biological. What would ammonia-based life on Titan produce? Different methane isotope ratios, anomalous acetylene depletion, unusual distribution of complex organics in the lake sediments. We're going to Titan specifically, so we have some shot at detecting this, only if the instruments are calibrated with the right hypothesis. If we're looking for Earth-style biosignatures, we'll generate reams of data about Titan's chemistry and miss the biological signal entirely. Biosignature detection missions cost billions of dollars. The Europa Clipper cost $5 billion. The James Webb Telescope cost $10 billion. Mars sample return is projected at over $10 billion. Every one of these missions was designed with a specific chemical target in mind. If the chemistry of life on those worlds is fundamentally different from our target chemistry, every one of these missions could return negative results and we'd declare those worlds lifeless. We might already be doing this. Every negative biosignature result from every world we visited has been interpreted through a carbon-centric lens. Titan, complex chemistry, but no life confirmed. Enceladus, organic molecules, but no life confirmed. Mars, perchlorates and methane anomalies, but probably geological. Here's a useful thought experiment. Imagine an alien civilization that evolved in an ammonia-based chemistry. Their instruments detect ammonia biosignatures. Their definition of life is built around ammonia-based metabolism. They fly past Earth and scan our atmosphere. They find an atmosphere almost devoid of ammonia. Their biosignature instruments return negative results. They declare Earth lifeless and move on. We would have been missed. 8 billion humans, a trillion microorganisms, the greatest known concentration of biological complexity in the galaxy, missed because the instrument was looking for the wrong molecule. This is not a hypothetical failure mode. It is the operational default of every astrobiology mission launched in the past 50 years. And the root of the problem isn't the instruments. The instruments are fine. The root of the problem is the definition, what we mean when we say life. Because if we can't define life without reference to our own chemistry, we will always design searches that find only our chemistry and call everywhere else dead.
Here's a question that sounds simple and turns out to be an intellectual abyss that has swallowed philosophers, biologists, and at least 2 Nobel laureates. What is life? Life reproduces. So do crystals, growing from supersaturated solutions, producing daughter crystals with identical structure. Not alive. Life metabolizes, takes in energy from the environment, and uses it to maintain internal order. So do candle flames, which consume wax and oxygen and maintain their shape and internal temperature in dynamic equilibrium with the environment. Not alive. Life responds to its environment. So does a thermostat. Not alive. Probably not alive. We hope not alive. Life evolves through natural selection. So do computer viruses, in a non-trivial sense. Viruses also evolve. We argue about whether viruses are alive. We've been arguing about this since we discovered them. Every criterion we propose for life, when examined carefully, either excludes things we consider clearly alive or includes things we consider clearly not alive. The definitional project has been ongoing for the entire history of biology and has not yet produced a consensus answer. NASA's working definition, a self-sustaining chemical system capable of Darwinian evolution, is the most operationally useful definition available. It's just not philosophically satisfying. And more practically, it contains the word chemical, which immediately excludes plasma-based structures, mechanical replicators, and anything else that operates by non-chemical principles. If we replace chemical with physical, a self-sustaining physical system capable of Darwinian evolution, suddenly the definition expands enormously. Plasma structures that store information in electromagnetic configurations and compete under selection pressure. Von Neumann machines that replicate and evolve. Quantum systems, and here we enter territory where the physics gets genuinely speculative, that maintain complex information states and reproduce them. Now, here's the part of this documentary that will either blow your mind or deeply annoy you, depending on your philosophical temperament. Let's apply the revised definition to stars. Stars form from molecular clouds. They maintain themselves against gravitational collapse through the energy of nuclear fusion, a form of metabolism in the broadest sense. They evolve over time, changing their structure, composition, and output across billions of years. And when they die in supernovae, they seed the surrounding space with heavy elements that become the molecular clouds that form new stars. Stars, in some sense, reproduce, not by dividing, not by copying genetic information, but by contributing to the formation of successor objects. Different types of stars produce different nucleosynthetic outputs. High-mass stars fuse heavier elements than low-mass stars. The specific properties of a star, mass, composition, metallicity, affect what elements it produces and seeds into the next generation of star-forming clouds. There is heritable variation. There is differential reproductive success in terms of contribution to the next stellar generation. There is arguably selection. This line of thinking was explored by Carl Sagan and others in the context of cosmic evolution. It's not a serious claim that stars are conscious or intentional. It's a more subtle point that the conceptual framework we use for life, heritable variation, differential reproduction, selection, might apply to systems operating at scales and by mechanisms we never imagined. And then there are black holes.
Stephen Hawking demonstrated that black holes are not perfectly black. They emit radiation through a quantum mechanical process at their event horizon, now called Hawking radiation. This radiation means black holes have a temperature, and they lose mass over time. They metabolize the vacuum. They will, over incomprehensible timescales, evaporate. Theoretical physicist Lee Smolin proposed a provocative hypothesis that black holes might give rise to new universes through their singularities, universes with slightly modified physical constants. If this is true, universes that produce more black holes would produce more offspring universes. Universes with physical constants hospitable to complex structure, and therefore to stars, and therefore to black holes, would be favored. This is natural selection at the cosmological level. Universes that are good at making black holes replicate. Universes that are bad at it don't. This is either profound or absurd, depending on your threshold for speculation. But notice what it requires: the same logical structure as Darwinian evolution. Heritable variation, differential reproduction, selection pressure. Applied to universes. The framework generalizes. It generalizes further than we designed it to. So where does this leave us, practically speaking? Because the philosophical horizon is interesting, but we have missions to plan and instruments to build and money to spend. The first shift we need is in how we define biosignatures. A biosignature is not a specific molecule. A biosignature is a pattern in chemistry, in thermodynamics, in information that cannot be easily explained without a self-organizing, energy-harvesting, replicating system. Different forms of life would produce different patterns. We need biosignature frameworks that are agnostic about chemistry, pattern-based, not molecule-based. This is not merely theoretical. Researchers at the Santa Fe Institute and elsewhere are developing life detection approaches based on information theory and assembly theory, frameworks that can, in principle, distinguish biological from non-biological complexity, regardless of chemical composition. These approaches would have looked at the Murchison meteorite's amino acid distribution and flagged it differently. They might have looked at the Viking labeled release data and reached a different conclusion. Chemist Lee Cronin at Glasgow University has developed assembly theory, a framework that measures the molecular complexity of compounds by calculating the minimum number of steps required to construct them from basic building blocks. Biological processes produce molecules with far higher assembly indices than abiotic chemistry, because biology has memory. It builds on previous constructions. Assembly theory can in principle detect the signature of biological process in any chemistry without knowing what that chemistry is in advance. When tested on Earth samples, it reliably distinguishes biologically produced compounds from geologically produced ones. When applied to the Murchison meteorite, it found signals consistent with biological level assembly. The methodology doesn't care whether the building blocks are carbon or silicon or sulfur. It cares about complexity and memory, properties of life regardless of substrate. The next generation of life detection needs to be fundamentally agnostic, not look for water, not look for carbon, but look for complexity that exceeds what chemistry alone produces. Look for patterns that imply memory, replication, and selection. Look for the shadow of process rather than the specific molecule. And with that framework, every world looks different. Titan's hydrocarbon lakes are not a dead chemistry experiment. They're a 4-billion-year-old chemistry experiment that we've never properly interrogated with the right questions. Io's sulfur volcanoes are not just geological spectacle. They are a sustained, energetically rich environment where complex chemistry has geological timescales to organize. The interstellar medium, the thin gas and dust between stars, contains complex organic molecules detected by radio astronomy. We find amino acid precursors, sugars, and molecular rings in molecular clouds light years across. Life, in the agnostic sense, may not be confined to planetary surfaces. The chemistry of life, at the minimum, operates at galactic scale. And here, from our small vantage point on this specific carbon and water world, we face the most humbling possibility of all. We have, for the entire history of our science, been looking in a mirror. We found organisms, we described what they do. We called that the definition of life. We built a universe-spanning search program around finding more of ourselves. It may be the most expensive case of parochialism in the history of intelligence. The universe has been running chemistry experiments for 13.8 billion years in plasma and ice and acid and vacuum and pressures we can't replicate and temperatures we can barely model. If self-organizing, energy harvesting, information storing, replicating complexity is an attractor state of physics, if complexity, given time and energy, is what the universe does, then we are not the exception. We are not the lucky accident. We are one solution among many. And if that's true, if the universe is not empty, but simply speaking in languages we haven't learned, then the silence we hear in the cosmos is not absence, it is incomprehension. We have built extraordinary minds out of carbon and water. We have used them to look at a universe of almost unlimited scale and diversity, and we have spent most of that looking time asking, is there anyone else out there who is basically like us? The better question, the harder question, the one that the evidence is increasingly forcing us to ask, is this: what if we are surrounded by life? What if life is what the universe is doing in 1,000 different chemistries and substrates and timescales, and we are simply blind to all but one of them?